So Keith Barker is an associate professor of ecology, evolution, and behavior at the University of Minnesota with an expertise in avian phylogeny and genetics, evolution of social behavior, spatial and temporal patterns, and molecular evolution. His current research focuses on patterns and processes of evolution in passerine birds. Keith.
Thank you for coming to the session today. I'm going to talk a little bit about some ongoing research that's happening in the lab right now. It's actually something I'm working on writing up at the this very moment. And this is a collaboration with my student, Tyler Imfeld, who's now faculty at Regis University in Colorado. And I'm listing him as first in this talk because he's actually the first author on the paper. We have a number of other collaborators on the work that I will mention at the end. But to place us in the broader context, in my lab, we're primarily interested in passerine diversity. And Passeriformes is one order out of about 30 orders of birds that contains more than half of the species diversity of birds. It's kind of ironic. I actually have a student, Shanta Hejmadi, who's studying raptors right now. So we're actually studying both ends of this. But today I'm going to focus on the passerine birds. And we've done a lot of work at the higher level relationships of passerines, but we're also sort of focusing on particular biogeographic regions. And one of the areas that we're really interested in is the Western Hemisphere, where we all currently are. So of passerine birds, slightly under half of all passerine birds are found in the Western Hemisphere. The remainder, of course, in the Eastern Hemisphere. And within the Western Hemisphere passerines, about half of those belong to two different groups. I'll say groups in quotes, because one of them is more or less a monophyletic group, the sub-Aucenes. These are things like flycatchers, manakins, cotingas, and allies, which are an autochthonous radiation that has its center in South America. And then this other group here, the Aucene passerines, is a melange of lineages that have dispersed from the Eastern Hemisphere to the Western Hemisphere throughout the last 10 or 20 million years. And so one of the interesting things that gives us the opportunity to do is to study the diversity of those lineages and how successful they've been in achieving an array of morphologies associated with feeding, as well as species diversity, and also the processes by which they disperse between areas within the Western Hemisphere. Yeah, so that's what I'm talking about there. We're focusing on that group, the Western Hemisphere Aucenes. And of course, some of the basic questions we have is, how did this diversity get here? When did it get here? And how successful have different lineages been once they've arrived here? And I'm going to be a little didactic today. We're going to talk a little bit about sort of broad scale approaches to understanding these kinds of questions before I get into nitty-gritty of a particular group. Broadly speaking, we study diversity, the history of diversification of different groups, sort of with two major sources of information, one of which is the fossil record, and one of which is phylogenetic inference, so understanding how species are related to one another. And it's pretty clear how we use fossils to study the history of diversification. So we've got a clear time component. So horizons that are deep in the sequence are older, and horizons that are on the top of the sequence typically are younger. And we can look at things like diversity. How many different species are there at different time slices? So we have a lineage that appears in one time slice, it diversifies into many lineages, and then maybe declines over time so that relatively few members are currently extant. We can also get spatial information from the fossil record. So if we look at the distribution of species, so if we have continents represented here by rectangles, we've got a fossil that may be found in multiple continents. We can look at both the time and the spatial components of the fossil distribution and say that a lineage may have arose in one place, say the red continent, dispersed into the blue continent, and was widespread at some point, and then went extinct subsequently in its area of origin, the red continent. So This is a classic type of evidence that's been used for many years to understand the history of, for instance, mammal diversification. And here's an example from the Great American Interchange, which is what happened around 3.5 million years ago, when South America, which had been isolated from all other land masses through most of the Cenozoic, suddenly came into contact with North America with the closure of the Panamanian Isthmus right here, so that species that had previously been prevented from dispersing between the continents because they weren't very good at dispersing over water, suddenly could move between those areas, right? And one example that biogeographers always love to point out, for instance, is that South America has 2 species of camels, the guanacos and vicuñas. But North America, as you probably know, currently has no camels native to it. But it's actually pretty clear from the fossil record that the camelids had their origin in North America, dispersed from there to South America as well as to the Eastern Hemisphere, and subsequently went extinct in North America. So that's a pretty clear way of inferring that biogeographic history. And that's possible because mammals are big, and they have things like teeth that are very identifiable, and it's so really easy to, in many cases, to track that history using fossil data. Birds, on the other hand, are small. They have delicate, fragile skeletons. Modern crown-clade birds don't have teeth. The fossil record, consequently, is very poor for a lot of bird lineages, and it's really hard to get that level of detail on things like biogeographic history. So for a lot of these bird lineages, we're depending on phylogenetic information. And so just to orient you to what a phylogeny is, we have species here at the tips. Here I'm labeling them with letters A through F. And then time flows from the root of the tree to each of these extant species at the tips. And then we have common ancestors that occur at these nodes interior to the phylogeny, right? And this is a genealogy of species. So strictly, it's directly comparable to a genealogy, say, in animal breeding or in human families, where we have tips, in this case, are individuals that are currently alive, perhaps, who have parents and grandparents who represent ancestors, common ancestors. Just like in a phylogeny, we have tips, which are species, and then we have interior nodes that represent ancestral species, which typically we haven't observed, especially if we have a lineage with poor fossil record. But even with lineages that have great fossil records, it's often hard to know if you really have an ancestor. So how does this tell us about biogeography? So here's our rectangles again. I've got species A, B, C, D, E, and F. A and B are found in the red area. D, E, and F are found in the blue area, and C is widespread in both areas, right? We can take that phylogeny, we can replace the current distributions of those species on the tips and actually think about them replacing those tips, and then we can come up with scenarios. Let's imagine the common ancestor of this whole group was found in the red area. We might imagine that it dispersed a number of times, in this case 4 dispersal events, to reach its current distribution. Right? So that's one scenario to explain the current distribution. By contrast, if the lineage was blue ancestrally, we might see that there were only 2 dispersal events that are required to explain its current distribution. So under Occam's razor, the principle of parsimony, we might actually prefer this scenario to this scenario because it requires fewer evolutionary events. And so this, in a nutshell, is the kind of analysis that we do when we take a phylogeny and current distributions of species and infer where those lineages might have been in the past. We look for histories that maximize the probability of observing what we see currently in terms of distributions. And usually that happens in a probabilistic framework, which is a little more complex than the simple parsimony example that I gave, but it's the same idea. So this is what we did a while ago now with the phylogeny of higher level phylogeny of all passerines. And what we found was that this one lineage, the Ossine passerines, actually was ancestrally Australasian and dispersed multiple times out of Australasia with especially one large group that we call the Passerida, dispersing out of Australasia and diversifying extensively across the northern hemisphere and then back into Africa, actually. And a number of those lineages dispersed from the green area here, which is Eurasia and Africa, into the blue area here, which is the Western Hemisphere. And you can see that I've got three specific lineages picked out as dispersing between the Eastern and Western Hemispheres. And we've been really studying these lineages as well as in quantifying how many lineages there are in order to understand how those lineages have diversified. Based on the time scale for this phylogeny that we've inferred, we can actually say that most of the lineages that made it from Australasia into Africa and Eurasia into the Western Hemisphere probably made it through one of these three routes. So here's a Beringia, here's a North Atlantic, and here's a southern route through Antarctica. But because of the recency of most of these dispersal events, we think that the primary route was probably across Beringia, when the climate there was much more favorable, and at times when there was a land bridge between the Eastern and Western Hemispheres, right? So the question is, how many times have these lineages made it across? How diverse are they? And then how did they get between North and South America? How often did they do that? And how species rich are those lineages? So this is Tyler, who's out in Colorado right at the moment. And this is some of his dissertation work, which is currently in review. We looked at quantifying how many different lineages made it from the Western to the Eastern Hemisphere, and of Aucenes in particular. And there are 44 separate lineages, 28 of which have more than 2 or more species in them, right? And the cool thing for Tyler's dissertation work was that we had phylogenies for almost all of these lineages, which is useful to do the kind of work we're interested in. But what wasn't so cool for his dissertation work was that there were no phylogenies for 3 of those lineages, right? And actually 3 of the most species-rich lineages that we have. One of which is the Embryozoidia, which has 824 some species. And that's a work that was finished just before Tyler came into the lab. And we now have a complete species-level phylogeny of Embryozoids, which I'm not going to really talk about today, but we'll actually use that for a point of reference later. Another of which was the Chloriphonia euphonia lineage, which turns out the phylogeny of that was a chapter of Tyler's dissertation. And then the last one is the one I want to talk about today, is the phylogeny of the Troglodytidae and the Polyptylidae, which is the second most species-rich lineage in the Western Hemisphere of species-rich lineage of Aucenes in the Western Hemisphere, and is actually the oldest lineage of those. All right? And I think I've spoken to you before and said that Embryozoidia is a great example of lineages in the Western Hemisphere because it looks like a good candidate for being an adaptive radiation. It's really species-rich. So here we've got the age of clades versus their log diversity. Embryozoids are really much more species diverse than we would expect given their age. Wrens, on the other hand, are down here. They're just about as species-rich as we'd expect given their age. So they're maybe more of a vanilla radiation, more a typical radiation of something that a lineage that has gone from the east to the Western Hemisphere. But we're going to study what's happening with those guys. Whoops, jumped ahead. So what do? We assembled all previously published sequence data for Certhioids. And this was at least 24 different studies that have been done on various groups of Certhioids. So the lineage, I should point out here, this lineage is just a subset of the group. This is the Wrens and allies, which what that means I'll define in a little bit. The lineage we actually worked on that I'm going to talk about is a broader, slightly broader lineage called Certhioidea. It includes a number of other groups. So we integrated those 24 studies, all the data from those 24 studies, along with new data for all but 2 previously missing taxa. So we've got data in this study for all but 2 species in that group, which is pretty good. We've analyzed all the data in a likelihood and Bayesian framework to create a time-calibrated tree for this group, a phylogeny. And then we did biogeographic analyses to see what insights we could get into when this lineage got into various parts of the Western Hemisphere, especially when it moved between North and South America, the kind of analysis that we did with fossils for mammals, right? And here's this phylogeny. This is a phylogeny. You don't expect to read all these little labels. We're going to walk through the major groups here. So this is 147 taxa. It's a concatenated analysis if there are any phylogeneticists in the room. We used 11 genes from 9 loci, including 3 mitochondrial and 8 nuclear genes. And so let's walk through the tree. So this is the monophyletic Certhioidea. The sister group to all other Certhioids is this guy, the wall creeper, Ticodroma muraria. And I tell you, I'm very sad that I've never seen one of these things in life. They live in Eurasia. I guess it's pretty common to see them on churches in parts of Europe in the wintertime. Beautiful, beautiful bird. Sister to all of their Certhioids. The next group here is this clade, a fairly small clade of nuthatches in the genus Sitta, including our nice friend, the white-breasted nuthatch, is an exemplar. There are some really cool blue ones from Southeast Asia as well. And then there's a clade of creepers. And there's 2 major groups of creepers. There's the spotted creepers in the genus Salpornis, right here. And that has 2 species, one of which is in Africa, East Africa, and one of which is in India. Widely disjunct genus, which is pretty interesting distribution. And then the true creepers, genus Certhia, they're distinct because they have this recurved tail feathers that they use to prop themselves up, which Salpornis doesn't actually have, including our good friend Certhia americana. And then this last group in the radiation is the Wrens, Gnatcatchers, and Gnatwrens. And this is the group that's present here in the Western Hemisphere. And Tyler and I spent so much time talking about this clade, it doesn't have a name. It's kind of useful. It's a monophyletic radiation of birds in the Western Hemisphere. And we said, we talk about it, anything that we talk about typically as phylogeneticists, we want to give it a name so we don't have to say Wrens, Gnatcatchers, and Gnatwrens every time. So we put our heads together, and the best we came up with was this group here called the Orthoura, which means upright-tailed, right, from the Greek. And, not all wrens and gnatcatchers have upright tails, but enough of them do that I think it's probably a legit way to describe this group. So the Orthoura is the Western Hemisphere members of the Certhioidea, right? And within that, there's 2 major groups. There's the gnatcatchers and gnatwrens. We still haven't named this group. We don't have to name this group. This is the Polyoptilidae, the family Polyoptilidae. Polyoptila, Microbades, and Ramificanus. Right here, this is a Microbades. Microbades and Ramificanus are tropical species. And then finally, this large group, the most species-rich lineage in Certhioids is the wrens. And these are Cynocerthia wrens from the high Andes in South America. Really cool birds live in these really large flocks and chorus and cooperatively breed and do all kinds of great stuff. Let's see here, where are we at? Right, so we have a phylogeny. We know major groups, we've named a useful clade. What about the biogeography of this lineage? So here we are. This is putting that tree together. So here's the tree, and then we've got the current distributions. That's what these are up here. So these would be our red and blue rectangles that I showed you in the example, plotted at the tips of each of the species. And then these circles back here on the nodes are inference about where that lineage most likely was at some time in the past. So these represent what we call marginal likelihoods. That's kind of the strength of support for alternative distributions at those various times in the past, right? So if you look at the root node for this whole lineage, the Certhioidea, you can see most of the circle is yellow, which means that there's a majority of support for that lineage having its origin in Eurasia, which makes sense. The Passeridae had their origin here in Australasia. They most likely dispersed into East Asia, although some people have actually argued that they jumped straight across over to Africa. don't really put much credence into that hypothesis. But they radiated here and then made it over into the Western Hemisphere, right? And then a lot of that early history is still in Eurasia, right? Especially for nuthatches and creepers. Although creepers obviously have made it in, and nuthatches have made it in a couple, 3 times into the Eastern Hemisphere or Western Hemisphere. And then this monophyletic group of Western Hemisphere critters, the Orthoura, you can see it's about 3/4 of the marginal likelihood is dark blue, which suggests that this lineage had a North American origin, right? So some ancestor here dispersed from the Eastern to the Western Hemisphere and founded this Orthoeuran lineage. And probably its early diversification happened in North America, but you can see pretty quickly this group here is the Polyoptilidae, probably made it into South America pretty early, whereas the Wrens, which is represented here, diversified for a while in North America prior to subsequently dispersing into South America a number of times, as you can see here. So the group made it, as we hypothesized, across Beringia into North America, diversified in North America a little bit, and then both Polyoptilids and a number of Wren lineages fairly quickly made it into South America. And then we've also got some evidence of back dispersal back into North America in some of these lineages. So what about this question about the Great American Biotic Interchange? We have all this great fossil data for mammals. What does this have to say about interchange for birds, right? Here's our cute little wrens. How many times have they been going across the isthmus? We already know something about directionality. I told you this group had its origin in North America, most likely. If you look here, I want you to focus on this panel down here. So this is an analysis of dispersal events between North and South America for the Orthoura. And this is showing the density of lineages that cross the Panamanian Isthmus as a function, or controlled for the number of species that existed at a given time in history. And what you can see is the highest probability, relative probability of crossing the isthmus was actually back here before the shaded area. And there's not much change across this shaded area. The shaded area is our estimate for when the Panamanian Isthmus became subaerial, that is, became land, dry land. So it doesn't really look like the Orthoura responded to the closure of the isthmus. Some things got in early, like the gnatcatchers. But after that, even though the lineage as a whole was crossing back and forth, there was not any detectable huge flush of dispersal. And this was pretty shocking to me, actually, because we did the same analysis back in 2015 for the Embryozoids. And you can see here's that gray area again, and here's the probability of dispersal. There is a really huge uptick in dispersal across the isthmus after closure of the isthmus in Embryozoids, which is, it's pretty strongly contrasting between the Embryozoid result and the Orthoeuran result. So it looks like Orthoura, for whatever reason, even though they tend to be much more forest interior species and less sort of scrubby growth, secondary growth habitat species, don't show an effect of closure of the isthmus, whereas the lineage of Embryozoids, many of whose species do live in secondary scrubby habitats, so that you might get when you first have land emerging from the ocean after closure of the isthmus, does show a strong effect of isthmian closure. So we got to put that in the hopper and think about that for a while, because I really wasn't expecting this kind of contrast in results between Embryozoids and Orthoeurans. Part of it might be just that there's so many fewer Orthoeurans, like a factor of 8 relative to Embryozoids, and we don't have the power to detect that. Or it could be something else that's going on that's really interesting. So we've just barely started thinking about that. And I think it looks like I'm about out of time, so I'll just summarize. The superfamily Certhioidea includes major Western Hemisphere radiation that we have now named the Orthoura. That includes the Polyoptilidae and the Troglodytidae. So if you ever want to, this name is not published yet, so maybe you don't want to start throwing it around in casual birding conversations, but If you ever want to talk about the gnatcatchers and wrens together, you could call them orthoerans. That lineage is the oldest and second most diverse radiation of Western Hemisphere Aucenes. And while it arose most likely in North America, which is actually something that Ernst Mayr predicted back in 1940s based on his knowledge of their distributions in biology, many of those lineages dispersed into South America. But unlike the Embryozoids, which we previously analyzed, there's no real signal of increased dispersal between North and South America after the closure of the isthmus. So for some reason, the wrens and their allies don't seem to be as sensitive to that closure as the Embryozoids were. It may be that I'm thinking about it wrong, and that since they are more dependent on these primary habitats, that they weren't able to take advantage of that early the formation of habitats when the isthmus was first formed. I'm sort of thinking out loud in front of you right now. But anyway, if you have some ideas, I'd love to hear it. And I think, yeah, it's about 26 minutes, so I'll stop right there. Oh shoot, I guess I'll throw up my acknowledgement slide, which I forgot to do, while people ask questions if they have any.
From somebody who flunked biology, is there any, do you have any speculation or is there information about the dispersal from Asia to North America of birds? Did birds follow animals? Other animals follow birds? What do we know about that?
Well, that's a good question. And presumably, Beringia was kind of a filter. The habitats weren't ideal for a lot of lineages of birds, mammals, and plants. I don't know of any interaction between different animal lineages that may have facilitated or prevented dispersal, like one dispersal of one lineage or another. Most likely, the environment itself was a filter to those lineages that made it through. And also, I think there are probably pretty significant secondary filters in Western North America. So there's a lot of cool lineages that have just got to Western North America and have just sat there and not diversified. So like the wren tit, the Orthoera, which is the verdin, they're one-offs, and they made it, but they haven't done much since they've gotten here. And probably because they got stuck in Western North America and haven't made it into the richer habitats further east in North America or south into the subtropics.
Does that mean that we're stuck in the rich poor area of North America here in Minnesota?
Well, we're kind of on the southern edge of that really rich forested habitat, which has promoted the rapid diversification of things like wood warblers. We have huge radiation of wood warblers that was supported by these environments. So,
Thank you so much.
thank you.